Alright folks, we're going to begin the details on some of these organelles. Alright, the cytoskeleton has three cytoskeletal elements. In other words, three different types of proteins that make it up. And those three proteins, from big to small, are microtubules, intermediate filaments, and microfilaments. So the microfilaments are the small ones. They're only about 7 nanometers diameter. The microtubules are the big ones. They're about 25 nanometers diameter. And then the intermediate filaments fall in the center. They're about 10 nanometers diameter. What microtubules do is they are involved in mitotic division. They pull the chromosomes apart. What microfilaments do is they do a lot of things actually. They are help with the shape of the cell. They help with the movement of the cell. They're involved in muscle contraction in a muscle cell. They help form microvilli. So they do quite a bit. And in cell division, they also are what does cytokinesis. The intermediate filaments are involved in keeping the organelles in place. They make up this network in the cytoplasm, and the organelles are attached to it. So that's the primary protein of intermediate filaments, by the way, is keratin. The same keratin that's in your fingernails and skin and hair. But it's not the only protein that makes up intermediate filaments. It just happens to be one of them. So keratin is a protein found in microfilaments. The cytoskeleton, especially actin or microfilaments, is involved in making microvilli, and you can see that here. Microvilli, of course, increase surface area for absorptive surfaces. The cytoskeletal elements, namely microtubules, make your cilia. And remember, from the centrosome, also called the microtubule organizing center, in the centrosome or the MTOC, we have centrioles, and from the centrioles we grow these microtubules that become cilia or flagella. Eukaryotic cilia and eukaryotic flagella have some architecture that's predictable. And if you look at the centrioles, you see 9 triplets. If I start right there and count 1, 2, 3, 4, 5, 6, 7, 8, 9, that's 9 triplets. And there's nothing in the center, so we call that a 9+0 organization. But as you start growing microtubules out of this centriole into a cilia or flagella, the 9+0 microtubule changes. And it changes at the area we call a basal body. So right here is the basal body. Below the basal body, I'm 9+0. But above the basal body, I'm 9+2. And it's not 9 triplets anymore, it's 9 doublets. So if I start right there, I have a doublet right here. 1, 2, 3, 4, 5, 6, 7, 8, 9. And plus I have 2 in the center, so that's a 9+2 arrangement. So my centrioles, right to my basal body, I have a 9+0 triplet organization, 9 triplets plus nothing in the middle. And above my basal body, my microtubules change over to 9 doublets plus 2 in the center, or a 9+2 arrangement. And those arrays are predictable in eukaryotes, but flagella and prokaryotes are different than that. The ER comes in 2 flavors. There's rough ER, which does protein synthesis. The rough ER is rough because it has ribosomes on it. You can see all these ribosomes studded on the ER. And the smooth ER is smooth because it doesn't have ribosomes. The rough ER does protein synthesis because ribosomes do protein synthesis. The smooth ER does a couple things. It does lipid synthesis, and it also detoxifies. It also stores calcium for us. So the smooth ER and the rough ER have different functions. I want to show you something here because later on I'm not going to be able to see it. Do you see how the nucleus is well, quit doing that. The nucleus is continuous with the rough ER. You can see it a couple different places. So I wanted to show you that because a little bit later I'm going to mention to you that the nucleus is continuous with the rough ER, and it's not going to be clear. So I want to show you that right there. The Golgi apparatus packages and modifies proteins. It's made of phospholipid bilayer membranes, and it packages and modifies proteins. It also does carbohydrate synthesis. And a Nobel Prize was just awarded a couple years ago for vesicular trafficking. And what that means is, how do we know where these vesicles are going? See, these vesicles pinch off the Golgi, and they have the protein that's been modified inside it. Well, where's it going? What's it going to become? Does it stay a vesicle? Does it become a lysosome? Is it destined for exocytosis? How do we know? How does the cell know? And the answer is now being figured out. And we're awarding Nobel Prizes now to research groups that are figuring out this vesicular trafficking. By the way, it all goes back to chemical signals. Lysosomes do intracellular digestion. And what I mean by that is a lysosome, like you see right here, can fuse with something like a phagosome or an endosome. Can't spell. Endocytosis, or a special type of endocytosis, is called phagocytosis. So when you do phagocytosis, that is called a phagosome. If you do endocytosis, it's called an endosome. Either way, phagosome or endosome, fuses with the lysosome, and the acids and enzymes of my lysosome digest that, and I can get rid of the waste product. Likewise, my lysosome can fuse with a damaged or worn out organelle, and I can digest that, and I can get rid of the waste products. Likewise, this lysosome could be involved in killing the cell itself, something called, well, autolysis is when the lysosome breaks down and releases digestive enzymes. That's autolysis. But doesn't that hurt the cell? Well, it could, but it could be programmed. And programmed cell death is called apoptosis. Now, we hope it's programmed, because if you kill a cell in an unprogrammed, uncontrolled fashion, that kills neighboring cells, which kills neighboring cells. And you see where I'm going with that. It's just a domino effect of killing cells. And that leads to inflammation, and that type of cell death is called necrosis. Death of tissue or death of cells is called necrosis. But apoptosis is the death of cells as well, but it's programmed cell death. It's controlled and stepwise and doesn't lead to a domino effect. But the lysosomes are involved in that.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
Okay, so that's the lysosomes in a nutshell. This is called the endomembrane system, and actually the video just before showed you that. When you make proteins, like enzymes, what happens is your DNA codes for messenger RNA. Here's a messenger RNA. Now the messenger RNA leaves the nucleus and either sits on a free ribosome or sits on a RER, rough ER ribosome. Either way, you do translation and you make your protein. Now free ribosome protein synthesis is usually making proteins that stay inside the cell. Whereas rough ER protein synthesis right here is making proteins which are destined to be exocytosized or become part of another organelle. So they might stay inside the cell, but they'll be packaged in another organelle. And this whole process, going from nucleus to rough ER to transport vesicles to Golgi, being modified and packaged into vesicles or organelles and either staying in the organelle or exocytosized, is part of the endomembrane system. So this is not willy-nilly and random. We have inside our cells this endomembrane system that passes off products to the next steps until you ultimately get your finished product. So it's the endomembrane system. Peroxisomes are organelles. We find them in plants, but we also find them in some human cells. They contain hydrogen peroxide, but they also detoxify hydrogen peroxide and other toxins. They neutralize toxins. We first found them in plant cells because way back in the day, before we had, you could go down to the salon and buy uh, hair products that lightened your hair. People used to use plants to add peroxide to their hair and lighten it, and it's the plants had these peroxisomes with hydrogen peroxide in it, so people used to use that. This is a drawing from Sandro Botticelli. It's a drawing of some people from Italy that this is an area of Italy where nobody had light hair, everyone had dark hair. But these folks were using plant products to lighten their hair, and that's the peroxisomes of the plants that gave them hydrogen peroxide. This is another one by Sandro Botticelli. And again, it's another uh, uh, Italian or Greek or something like that, some Mediterranean hair group that typically doesn't have light hair, but they would lighten their hair with the plant hydrogen peroxide from the peroxisomes. Mitochondria is the powerhouse of the cell. It makes 95% of your ATP. Mitochondria are double membraned. So here's one phospholipid bilayer, and here's another phospholipid bilayer. The aerobic respiration occurs in these mitochondria, and in chapter 25, I believe, we go through the details of aerobic respiration. So this picture right here is a really, really rough overview of metabolism. And glycolysis occurs in the cytoplasm, but Krebs cycle and electron transport chain occur in the mitochondria. Citric acid cycle is also known as Krebs cycle. And you make most of your ATP in your mitochondria. The nucleus is a double membrane system. This is one phospholipid bilayer here, and here's another phospholipid bilayer here. The nucleus has pores, and these pores are large enough to let messenger RNA and some small proteins in and out. Well, the messenger RNA out, the small proteins in and out. And we'll talk a little bit about that in the future. But the nucleus is a double membrane system which contains your DNA. And a darker staining area in the nucleus, right here, that's called the nucleolus or nucleolus. And that's an area of a ribosome assembly. So yes, the ribosome proteins are made in the cytoplasm because that's where the other ribosomes are, because the ribosome itself is made of proteins. But they're assembled right here, and then they go through the pore and set up house out in the cytoplasm. I'll see you in the next mini lecture.